So it's my pleasure to introduce the next speaker, Shay Salehi from GSK, to give us an update on the evidence for durable response with bepirovirsen.
Thank you. Good morning, ladies and gentlemen. Thank you, chairs, for the kind invitation to participate in this wonderful meeting. My name is Shay Salehi, and I'm Global Medical Affairs Director for GSK. And it gives me great pleasure to spend the next 10 minutes or so discussing some of the durable response data that we have to bepirovirsen in our BClear phase 2b study. And I must start by saying that as a clinician, the last few days has been quite complicated scientifically, but it's great to see so many people focused on hepatitis B research. And functional cure is a really an elevated treatment goal for people living with hepatitis B. So it's a really exciting space to be working in. So here are my disclaimers. Bepirovirsen, or Bepi as we like to refer it, is an unconjugated antisense oligonucleotide. It is composed of a synthetic single-stranded single nucleic acid polymer with a mixture of DNA and RNA. And it's the chemically modified flanking RNA regions that promote target binding and protection from exonuclease. The central DNA gap permits formation of the DNA-RNA hybrid, and this allows the recruitment of RNase H that is present within the cytoplasm and nucleus and allows the degradation and catalyzes the target RNA. And this in turn silences gene expression. Bepirovirsen targets a highly conserved region across HBV genotypes and present across all HBV RNAs, including pregenomic RNA. This leads to three effects. So firstly, we have a reduction in viral proteins, including surface antigen, which we know plays a critical function in immune evasion and facilitates disease chronicity. Secondly, we have a reduction in viral DNA, thereby preventing the formation of new infectious virions. And finally, we have some preclinical data that suggests there's an immunostimulatory effect through mechanisms such as activation of toll-like receptor 8 pathways in the non-parenchymal cells, which we believe will support the revival of the chronic hepatitis B infection-related immune exhaustion. The effects have been investigated in a phase 2b clinical trial called BClear. The results were published in the New England Journal of Medicine at the end of last year. Now, this was a phase 2b clinical trial that looked at various doses and durations of bepirovirsen in participants on a background nuke, virally suppressed, as well as not on a background nuke. So approximately 460 participants were randomized in a 3:3:3:1 ratio into one of 4 treatment arms. Firstly, we have Bepi 300 milligrams for 24 weeks. Secondly, we have Bepi 300 milligrams for 12 weeks, followed by 150 milligrams for 12 weeks. Arm 3 included 12 weeks of treatment with Bepi 300 milligrams, followed by placebo for 12 weeks. And finally, group 4 had placebo for 12 weeks, followed by bepirovirsen 300 milligrams for 12 weeks. Now, the primary endpoint was HB surface antigen lower than the lower limit of detection and DNA less than the lower limit of quantification, sustained for 24 weeks following the end of Bepi treatment. Important to note that participants who were on a background nuke continued throughout the study, and the primary endpoint was achieved in up to 10% of participants in the overall population, but we saw a significantly higher number of participants achieving the primary endpoint in those with a low baseline surface antigen, up to 26% in those with a baseline surface antigen of less than 1,000. The ability to achieve functional cure is being investigated in our pivotal phase 3 trial, which is ongoing, called Be Well. However, we are looking to understand the proportion of participants who have already received Bepi in our phase 2 trials who firstly achieve functional cure and have a durable response. Now, in terms of functional cure, in the context of Bepi, we've allowed for a 12-week washout period to ensure that there is no influence of bepirovirsen, to ensure that we're fully confident that it's off of treatment. We also need to account for the background nuke, so there's a nuke stopping step that's incorporated. And this is being investigated in our ongoing phase 2b long-term durability study called BE-SURE. Here we will have complete and partial responders from our phase 2 trials, followed up for up to 33 months, and the functional cure endpoint is determined at month 3 of BE-SURE in the not on nuke population. So this is up to 12 months off of bepi treatment. And in the on nuke cohort, we would assess for criteria to stop nucleoside analog and follow up for a further 6 months to define the functional cure. Here I'm showing you the first interim data that was presented at EASL this year, and this is the complete responders within BE-CLEAR. Here we had 12 participants enter the trial, and of the participants where we have 3 months data, so of the 9, we have 7 that have maintained a response. Now at the 9-month time point of BE-SURE, so these participants have been off of bepi for up to 18 months, data were available for 3 participants and all 3 had maintained a response. So we can say that 7 out of the 9 have achieved functional cure, and that appears to be durable. Important to note, of the 2 participants that didn't maintain a response, they did not meet criteria to restart nuke, or to start nuke in this case. Now moving on to the cohort who were on a background nuke, we had 13 complete responders from BE-CLEAR, and 9 of the 13 stopped nuke at the 3-month time point. And where data were available at 6 months of BE-SURE, we have 7 participants, and 6 had maintained a response. And of the 4 participants, we have data up to the 9-month endpoint, the 9-month time point. All of the 4 participants had maintained a response and therefore achieved functional cure. And again, the 1 participant that didn't maintain a response did not meet the criteria to restart nuke. So this is early data. The caveat is we've got a small number of patients, but we'll be looking to present data as it does mature, as the participants have followed up for longer, as well as incorporating the partial responders from BE-CLEAR. We're very aware that chronic hepatitis B is a very heterogeneous disease. We also see that in the response to bepirovirsen, and we're looking to understand more about how these patients behave utilizing artificial intelligence and machine learning, really to convert the high-dimensional data that's being collected within BE-CLEAR and condensing that down to understand the multivariate interactivity and clustering those into sort of easy-to-understand subgroups. So here we have some data that was presented at EASL this year, showing 5 subpopulations that display heterogeneity response to bepirovirsen. The red are the complete responders, and we can see that there are 4 subgroups that are non-responders. So in the sense that your clinical trial will say a binary yes or no to response versus non-response, we're able to really pick out the subtle differences between subgroups. And this is the reason why within our phase 3 clinical trial, we're really focusing on the less than 3,000 surface antigen population where the durability seems to stick. However, we're using this information to help define our clinical trial strategy so that the right patient receives the right, potentially the right combination at the right time. And we've got an extensive clinical development program that helps us to unpick the heterogeneity and response even further, but also looking at the impact of bepirovirsen as a monotherapy in the right patients, but also as a backbone to combination and sequential therapies in those that to increase the functional cure rates in those that have a lower surface antigen, but also increasing the number of patients that may benefit from bepirovirsen, for example, those with a greater than, with a high baseline surface antigen. So I will stop there. I just want to close by thanking our collaborators, our investigators, and of course our patients, without who we would not have a clinical trial program. Thank you.